is on quorum sensing, the concept of cell-to-cell communication mediated by small signaling molecules. In gram-negatives, they're N-acyl homoserine lactones. In gram-positives, it's a little bit different, as I'll show you in a minute. Essentially what happens, though, is the cells are coupling cell density and intercellular communication to transcription regulation. So here's a side-by-side look at gram-positive and gram-negative quorum sensing systems. On the left is the gram-positive quorum sensing system, and in this particular case, the microbes will make a peptide signal, so it's a short amino acid. As you can see down here, here's an auto-inducing peptide from the gram-positive bacterium Staph aureus. Well, here's two of them, auto-inducing peptides, and so it's this short little chain of amino acids right here that bends back and binds to itself, this little disulfide linkage right here. This peptide signal is produced into the world, and the microbes themselves can process that signal, or it can diffuse over to an adjacent nearby microbe. The microbe would have a sensor kinase in the cytoplasmic membrane that would recognize that auto-inducing peptide, which would then give it kinase activity to phosphorylate a response regulator, and in turn the response regulator could sit on a promoter and activate a series of genes. Over on the right is the prototypical gram-negative quorum sensing model, and again, it's not the only one, but I'll show you a few other variations. But in this particular case, the bacteria has a LuxI type gene, which is an autoinducer synthase. So here's a few different gram-negative autoinducers. They're these homoserine lactones with different side chains. So you have here's one with 1, 2, 3, 4, 5, 6 carbons in a R group of whatever. Here's one with 1, 2, 3, 4 carbons. So again, a C4 HSL and a 3-oxo C12 HSL, like this one here that Pseudomonas aeruginosa uses. So imagine this autoinducer synthase, depending on the organism, producing an autoinducer like one of these 3 types of molecules. This is the general structure, and these are more specific Pseudomonas autoinducers. So these signals are so small they can be produced and pumped out through the cytoplasmic membrane and out into the world, where they can either interact with a receptor inside the cell, or these little autoinducer molecules can activate or repress gene expression in a neighboring cell. And so typically there's a receptor of some kind, like a LuxR type receptor, that binds to the autoinducer, and that complex then can interact with a promoter to activate gene expression. Here's a more sophisticated look at it. This is the Vibrio harveyi quorum sensing model. And so in Vibrio harveyi, there's 3 systems. There's this 3 sensor systems, the LuxN system, the LuxP and Q system that you see here, and then the CQS system. The main ones that I want you to focus on are these 2, and there's another slide about these 2 down the road that I'll bring up. But anyway, LuxN is a sensor, and LuxP and Q are a combined sensor. And so you see LuxP just sticking out into the periplasm, and then LuxQ as this transmembrane protein. In either case, at low cell densities, none of the autoinducers is present in sufficient quantities to inhibit the kinase activity of the respective sensors. And so what that means is that there's no autoinducer, it's low cell density, or very low autoinducer, low cell density, and as a result, these guys have kinase activity, and they can pass their phosphate to a intermediate, a phosphodonor like LuxU, and then LuxU can pass its phosphate to an intermediate like LuxO, and then it's actually LuxO-phosphate in this format, in addition to RPON, this little guy. So this little complex here will activate small RNA formation, and then the small RNAs bind to HFQ, a concept you've already seen, and this complex acts as an antisense RNA system to shut down this major transcription factor transcript called LuxR. And so when that happens, you get no LuxR formation, and therefore it shuts down bioluminescence. At the bottom here, you see high cell density. And so there's there's various autoinducer synthases, like this LuxM that's synthesizing HAI1, so Harvey I autoinducer 1, and a LuxS type autoinducer synthase that produces autoinducer 2. So in system 1 here, autoinducer 1 will bind to LuxN and block the phosphorylation cascade. Similarly, autoinducer 2 here will bind to LuxP and Q and block its kinase activity, so blocking the phosphorylation cascade. So these guys are never phosphorylated, and as a result, LuxO never gets activated. So this repressor, LuxO, never gets activated, which in turn allows LuxR messenger RNA to be translated into protein and to activate bioluminescence in this bacterium. Don't worry about this on the side here. This is autoinducer mediated or quorum sensing mediated transcription regulation in another Vibrio fischeri, the microbe that I work on. And so in this particular case, there are 3 autoinducers, and produced by 3 autoinducer synthases. So LuxI is an autoinducer synthase, AINS is a synthase, and LuxS is a synthase. And they each make these unique autoinducers. And so LuxI, this one here, is the primary autoinducer produced by Vibrio fischeri. It's a 3-oxo C6 molecule. AINS makes a 3-oxo C8 molecule. I'm sorry, not a 3-oxo, just a C8 molecule. There's no oxo at this 3-carbon position, so it's not a 3-oxo. Just like this guy, 1, 2, 3. There's the oxygen. That's why it's 3-oxo. Anyway, so C8 diffuses over, and you see a pool of C8s over here, and it interacts with this sensor kinase, AINR, as well as this receptor, LuxR. And so C8 autoinducer and LuxR can weakly activate the bioluminescence genes in Vibrio fischeri. And that's why this arrow is drawn very thin. However, the 3-oxo C6, when that builds up, it's a little slower than C8. So C8 gets going sooner and primes the pump a little bit with weak expression. And then the 3-oxo C6 builds up, and it binds to LuxR and more strongly activates gene expression of the lux or luminescence operon. That's why this arrow is drawn much bigger and thicker. And the LuxS molecule that you see here diffuses over and will interact with LuxP and Q. So these are similar systems that you just saw with Vibrio harvii. This system, however, is not present in Vibrio harvii. So again, the concept is the same. The binding of autoinducer to these things blocks the phosphorylation cascade, whereas the so the presence of autoinducer leads to less phosphorylated LuxO. The absence of autoinducer in a low cell density environment means that these guys will phosphorylate this guy. The LuxQ and LuxAINR will phosphorylate LuxU, and then LuxU will phosphorylate LuxO. And LuxO together will activate these repressors that will block LightR. Blocking LightR means that you will not have the activator that's necessary, one of the activators of this LuxR protein here. And so the LuxR protein doesn't get made, and it can't recognize autoinducer, and therefore can't stimulate bioluminescence. This slide is some data showing you an important concept in quorum sensing called cross-species quorum sensing. And so what the researchers did in this particular case is created 3 mutants, BB120, BB886, and BB170. I'm sorry, BB120 is the wild type. I even put it over here so I wouldn't say that, and I did it anyway. Anyway, BB120 is the wild type, meaning that it has sensor system 1 and sensor system 2. And so again, to remind you, here's sensor system 1, and here's sensor system 2 in Vibrio harvii. BB886 is sensor 1 positive, but sensor 2 negative. So again, if we go back here, it's sensor 1 positive, so it has lux N, but it lacks lux PQ. And BB170 is sensor 1 negative, but sensor 2 positive. And so basically all they do then is grow up an organism. So here's Vibrio harvii BB120, the wild type strain. They grow this guy up in media, and then they purify any autoinducers as this guy belched into the medium. And then they expose these particular sensor strains to that cell-free extract and ask the question, how much light was made? And so in each of these cases, it's all percentage of the wild type. So not surprising if you grab all the autoinducers produced by BB120 and throw them into a medium containing the BB120 strain, it's going to light up and produce 100% of the luminescence that it normally would. In this particular case, you've got the sensor 1 plus, sensor 2 minus. You throw the autoinducers from this guy into here, it's 100%, and similarly over here it's 100%, meaning that there was a variety of autoinducers in here and they stimulated this sensor system to make a ton of light. And in this particular case, the autoinducers in here stimulated this sensor to make a ton of light. So each of these numbers then represents a percentage of luminescence relative to the wild type. So in this particular case, they take the cell-free extract of Vibrio harvii BB152, and then they expose it to the wild-type strain here with sensor 1 and sensor 2, and they only get 32% of the luminescence. So that tells you that it's definitely making an autoinducer that can stimulate these one or both of these sensors. You don't know which one, but for sure it's not very strong. Over here, this guy has sensor 1, but not sensor 2, and there's almost no luminescence. So that tells you that whatever this guy's making does not stimulate sensor 1, and most likely stimulates sensor 2. This is where the data get a little weird, and I may ask a question about this so you can interpret, but Vibrio harvii BB152's extract will actually overstimulate this particular strain in terms of luminescence. So something in that extract is really binding strongly to sensor 2 and causing a pronounced reaction. So again, and you can use that sort of logic to look at all these different strains and get a sense for which of these organisms produces which molecule to stimulate which sensor in these sensor strains. And so this concept of quorum sensing, as it relates to luminescence, is illustrated in this graph. So on the y-axis, you see luminescence per OD. So it's basically light emission per cell. And on the x-axis, so this is the y-axis, the x-axis is cell density. And so this is a great way when you plot your data, if you're doing luminescence or any kind of quorum sensing related activity, you plot it versus cell density, so you can get a sense for whether the physiological effect is dependent on cell density. And in this particular case, you can kind of see that it is. Here, the luminescence values per cell are high initially, and then they drop, and then at some critical cell density, everything shoots up, suggesting that the microbes had to wait till they got to a certain cell density. That is, the culture had to contain a certain amount of autoinducers in order for gene expression to turn on. These last two slides are a little bit of a departure from quorum sensing, but again, still sticking to the same vein of gene expression. So what we're looking at here is starvation signals and how they influence sporulation in Bacillus subtilis, particularly through transcription factors as well as alternative sigma factors. And so that's pictured here. So in this particular case, you have a sensor kinase, and there's a starvation situation. So imagine Bacillus subtilis in some nutrient-poor, dry soil, and it can't get the nutrients that it needs. And so the microbe would perceive that, and then the sensor kinase, these proteins kin A through E, would autophosphorylate and then transfer their phosphate groups to a transcription factor referred to as SPO0F. Spo0F would then pass its phosphate group to Spo0B, and in turn Spo0B would phosphorylate Spo0A, and this active transcription factor would stimulate sporulation, would basically sit on these sporulation genes, gene promoter, and activate all the sporulation cascade. What's important to note also, though, is there's a variety of sigma factors that are involved here, and I've indicated them here in this little cartoon. So sigma F and sigma E, for example. So F and E are essential for the early phases of sporulation, such as engulfment, and so you can see that here. So here's this 4 spore forming in this bacterium, and in order for that to become an internalized spore, you have to have engulfment. So this little septum and so forth sort of forms around the 4 spore and drags it into the cell. And then these later sporulation genes are activated by sigma G and K, as pictured here, that form the spore coat layers, for example. And so that's what you see here, these additional layers that form around the spore coat to give it its environmental persistence.